Hi, I'm Khalid, a member of the Neuroplicity team. Have you ever wondered how your brain learns something? Whether it's how to sing a song, play tennis, balance on a bike, or do a math problem, your brain learns and remembers thanks to synaptic plasticity, a process that occurs in nerve cells. Dr. Nicole Calakos at Duke University is one of the many neuroscience researchers seeking to understand how synaptic plasticity works.
My name's Nicole Calakos. I'm an associate professor in neurology and neurobiology at Duke University, and my lab studies synaptic plasticity mechanisms, and that's our way of saying how connections between nerve cells change dynamically in response to experience.
Neurons communicate with each other at sites called synapses. At any synapse, there's always one neuron sending a signal and one neuron receiving it. Synapses can get stronger or weaker depending on how actively the neurons are being used. If the first neuron sends many signals, then the receiving neuron will set up more signal receptors, and the communication between those two neurons becomes stronger. If, on the other hand, the first neuron stops sending signals to the second neuron, then the receiving neuron will start to take down some of the receptors, and that synapse will weaken. This ability of synapses to change in strength is called synaptic plasticity. As the synaptic connections in your brain change, those changes can manifest in your behavior when you learn new things.
We have some projects studying how do we form habits? How do we go from learning stimulus response, in this case, for example, pressing a lever for a reward, to a behavior that's kind of on autopilot and habitual. This can be a really positive adaptive thing. It allows us to get through the day and do the kind of essential everyday things we need to do that are healthy and adaptive for us, and we could think about other more important things. So we study that process, and then our disease models are some examples of when that goes awry. Compulsivity can happen in a lot of contexts. We think sometimes of drug addiction behavior, but also things like OCD, obsessive compulsive disorder. You're fixed in a behavioral routine and you're not able to break out of it. You're not able to realize this isn't healthy anymore. It's consuming all my day, or if it's even hurting me and causing injury to me or my family. So the way we went about figuring out this problem was to model habit and compulsion in mice and then look at the brains and see whether there were long-lasting changes that could predict their behavior. And this is a summary of what we found, in that we saw in both habitual mice and compulsive mice, there were long-lasting differences in activity that predicted behavior. If you think of each of these uptakes as an action potential, and the two different colors are these two different cell populations that have kind of a push-pull influence on behavior to do an action or not, we see that in response to the same stimulus, the brains are much more active, firing more in habit and compulsion, but there's some subtle differences, and we're looking to these differences as cues to where now you get into a problematic behavior and think about that as targeting for treatments. And one interesting thing we found is that it's starting to make us think that they would have more of a problem breaking a habit, because we also asked what were the long-lasting changes in mice that successfully suppressed one of the habits they learned. So these are the kinds of things that we're doing to give us clues on the next steps.
Dr. Calakos' lab focuses on this in a region of the brain called the basal ganglia. The basal ganglia are a collection of brain structures located under the cortex. They are responsible for a variety of functions, including controlling your movements. Damage to this part of the brain causes OCD and many other disorders that Dr. Calakos sees, including Huntington's disease, dystonia, and Parkinson's disease. So, by studying disease models, or lab animals that have symptoms or mutations resembling a human disease, her lab can better understand how such symptoms arise and how that relates to normal synaptic plasticity.
I'm really interested in synaptic plasticity, and I could study it anywhere, but I brought it all into the people that I take care of, so that I'm constantly thinking back and forth about synaptic plasticity and their particular problems.
For more information, visit Dr. Calakos and her lab online.